This is the strawberry DNA extraction lab that was completed on April 29th, 2009 for Mrs. Swear's science class. For this video, we're going to be reviewing what we did during this lab so that you can get a feeling and understand what DNA extraction is like. Before we go and do the lab, I'm going to explain to you a little bit about what happens during the lab and why we use certain ingredients. First of all, we are going to be looking at a strawberry cell, and a strawberry is a plant, so we're going to be looking at a plant cell. The first thing that you need to know, and this is a review, is that the cell in a plant cell has two layers, and we've talked about this before in class. The first layer is known as the cell wall, and this is what gives a plant cell its rigid shape and structure. The second layer, or the inner layer of a plant cell, is known as the cell membrane. Now, the cell membrane controls what goes in and out of the cell. Another layer that you need to be aware of is called the nuclear membrane. And this is actually something that you will learn later in 10th grade biology. But we do need to know about it for the purpose of this lab. It's also called the nuclear envelope. I'm just going to write membrane here. So as you can see, our DNA is located inside the nucleus, but we have to get through 3 walls in order to extract it. So the first thing we are going to do in our lab is we're going to actually mash up our strawberries in a plastic bag. This helps break up the cell wall. So I'm going to put these little marks here to show that it's broken up and that there are little holes in the wall. So that will be our first step. The second thing that we need to do is we need to find some way to get in through the cell membrane and through the nuclear membrane in order to get our DNA out. So we are going to be using a DNA extraction liquid that's made from dish soap, salt, and water. Now, the dish soap acts as scissors to kind of cut open the cell membrane. There's a chemical reaction that takes place, and so when we add the soap to the strawberries, it will actually create holes or cut apart this cell membrane. It will also cut apart the nuclear membrane. This allows the DNA to come out freely from the cell. If we look over here, the next step is now we have our DNA, but now it's in the form of a chromosome. If you remember from our lesson, a chromosome is a piece of DNA that's coiled or supercoiled up on itself. So the DNA is wound up really tightly, kind of like a thread on a spool. So we want the DNA to be in strands so that we can see it easily. But the DNA is in this X shape. So the reason why it's in this shape is because DNA is coiled around things called histone proteins, or just proteins. So in order to get the DNA into a strand so that we can see it, we need to cut these out, and that's when we apply the salt. You can also use meat tenderizer. Using these two things helps cut out the proteins and makes our DNA into a strand shape. So this is why we put salt into our DNA extraction liquid. So what we'll do is pour the liquid with the dish soap and the salt and the tenderizer into the bag and mash it for about 1 minute. Afterwards, you'll get DNA strands that are long and loose. And if you were to magnify that, lots and lots of magnification, you would be able to see a DNA strand, which is a double helix. Remember, we have sugars and phosphates that make up the backbone, and then in the middle we have our base pairs. So that's why we use the ingredients that we're going to use. Later on, we're also going to pour in alcohol to be able to see the DNA. That creates a layer that is less dense than strawberry juice so that the DNA can float up into it, and then